Well, I'd like to thank the organisers for the invitation to tackle this quite challenging topic against my esteemed colleague Pamela Shaw, who I'm sure will produce a very persuasive argument against this idea that the study of mice has been detrimental to developing therapy for amyotrophic lateral sclerosis. So I'm Kevin Talbot from the University of Oxford in the UK, and my conflicts of interest are there and my funding sources. But also I'll have to give you a disclaimer, which is that in my research I heavily depend on mouse models of ALS for drug discovery. So you might well feel that I'm a hypocrite for standing up and talking against the idea they may be useful. In fact, I'm going to talk about the idea that they may be useful, but they have not been useful up to now because they've not been used correctly. So here you can see just in the last year or two some publications from my group using novel mouse model that we have created and how we are trying to use that for drug discovery. So I think a good place to start is to remind ourselves about the complexity of ALS and its essence. What's the nature of ALS? Well, it's a late onset degenerative condition which arises through multiple age-dependent steps. So this paper from Amar Al-Chalabi and colleagues demonstrates that in order to get ALS, a number of conditions have to be met. And having a genetic mutation, for example, is not a sufficient condition for ALS. You require a number of somewhat obscure at present, but a number of subsequent biological lesions or hits to occur to trigger the disease. And these are dependent on aging. And so one of my key messages really is that ALS is not a disease of toxicity. By and large, it's a disease of loss of tolerance for an injurious mutation. And that's one of the things that we have to try and capture in our models. The other key concept is that ALS is a disorder of a system. And in that sense, it's very like Parkinson's disease and Alzheimer's disease. Contrary to those people who might take the view that the whole nervous system becomes disrupted in diseases like ALS, which is certainly gaining some kind of currency with a kind of prion-like mechanisms that are very fashionable. Contrary to that view, actually ALS is a very constrained disease. It affects a very specific system, one which has connectivity, both anatomical and functional. And it's within that system that the disease plays out and the clinical manifestations occur. So the very large number of upstream biological hits which can drive that converge on this vulnerable network and this vulnerable system. So that concept is really critical for our modeling of disease. So we have a condition which has a vulnerable network system. It has many upstream biological causes, and we have an age-dependent loss of tolerance. And inevitably, the complexity that we see means that multiple types of approach therapeutically are going to be required, and there is no one-size-fits-all approach. So again, another key concept in understanding whether mouse models have been helpful is to try and get away from the idea that they can model the disease in its entirety and therefore produce drugs from often a rather narrow biological niche to do with a particular type of mutation. We must get away from the idea that that can produce something which is going to really cover all aspects of the disease, since that point of convergence on that system, behind that are many, many biological processes, and drugs affect biological processes. So this system comes with a very rapid recent evolution. Rodents and humans are separated by at least 50 million years of evolution. And even our closest primate relatives, like the chimpanzee, about 5 million years of evolution, are separated by very marked differences in the corticomotorneuronal system, with more one-to-one connectivity and more fibers devoted to dexterity and other aspects of particularly human sorts of functions. So if we think that this is a human disease of a human system which has evolved in humans, or at least hominins, then we are going to have trouble modeling that in its entirety in a rodent model, which has a very different sort of motor system. in which some things are undoubtedly preserved. And for example, the neuromuscular junction has some aspects of preservation which make people focus on it and use it as a readout. But that may in itself be misleading. So a really terrific statistician called George Box, and he's talking here about statistical models, but you can apply this to any scientific model. If you make a model, then you have to be able to tell yourself that the model is wrong. But then you have to try and understand whether it might be useful. So if you take the view that you are modeling a thing like ALS in a mouse, and you don't ask yourself that question, is any of this useful? Then you are simply going to blindly proceed in a direction that is going to lead to distorted outcomes. So the only question is not whether the model is true, which is often what people try and focus on, and the answer is always no. The only question is the model going to produce something useful, something illuminating? So if we take the view that all our models are likely to be wrong, then I think we've compounded the error by deliberately, if you like, or knowingly created errors from the outset. And we've done this because it seemed expedient to do so. So let me be absolutely clear that I'm not being critical of those people who in 1994 set out to make a cDNA-based SOD1 model. I'm sure I would have done exactly the same thing, and subsequently I did indeed use that model in my lab. But I think with 25 years of hindsight now, we have to try and understand what we can learn from the errors that were part of that process. So there were some systematic errors. So obviously we've already said that this is the wrong species. Now, one simply has to acknowledge that and try and understand how one can use the wrong species to get some useful information about humans. And I think the key element for me is not to expect the mouse to contain all elements of disease, because you can't recapitulate the aging phenotype of mice. You can't recapitulate the particular architecture of the human motor system, which may be absolutely part of the quintessential element of ALS and why we get it. So if you use phenotype-driven approaches in terms of motor and behavioral phenotypes, this is very high risk because it may produce drug outcomes which are certainly modifying those phenotypes, but which do not in any way translate into humans. And I think that's been a recurring theme. So one has to be very careful about phenotypic screens in mice, which are gross motor or behavioral. We have systematically used the wrong genotype, meaning we have taken concatenated cDNAs, put them into the wrong part of the genome, and without the normal regulatory elements, and expected those to recapitulate the complex biology that we see in a human. And obviously, one can do this knowingly, but I think people do it and then they move on and they forget they've done it. And therefore, they are puzzled when they are looking at phenomena which are a consequence of unregulated gene expression and potentially off-target effects. So all mice strains used in the lab are highly inbred. And also, you then put in some form of ectopic genetic element into the genome randomly, there is a chance that you're going to disrupt some endogenous genes, and that's certainly been done by mouse models. Then people use promoters which are not relevant to the genes. So they don't use human promoters, they might use strong promoters to drive high levels of expression. They might use cell-specific promoters like Thi1 for neuronal postnatal expression. And that may serve a particular purpose because it may concentrate your mutant gene and protein in a cell of interest, but you'll lose that spatial and temporal expression, which is all part of how, in a complex mammalian system, a gene is tolerated and how that tolerance breaks down. And then you end up with very high levels of protein expression from early stages. So I don't think anybody would argue that ALS is a disease of protein overexpression. I mean, in Parkinson's, you get triplicated synuclein mutations that you can titrate the levels of synuclein to the chance of getting the disease. I don't think there are any similar examples in ALS. And yet, in order to drive phenotypes to produce mice that where things can be observed, where things can go wrong, people have chosen to use very high levels of expression, which are very unphysiological. And that produces an awful lot of harm. It may even produce elements of a disease that are highly relevant to the disease, but it gets swamped, in my view, with phenomena that are distractions and therefore do not produce translatable outcomes. These are very severe early onset phenotypes, which may be practical because you can turn around an experiment in a few months, but they don't reflect the disease as we see it. And lastly, as I've mentioned, this focus on toxicity rather than tolerance is problematic because it distorts the experiments towards those outcomes and phenotypes which can be measured as toxic insults. And these are either not part of a disease at all, or they are late consequences of many multiple upstream biological effects, which are the ones you really want to capture. So I think we make very unreasonable demands of mouse models. People will talk about a mouse with ALS, even more extreme, people talk about flies with ALS, and all of these statements need to be treated with extreme caution and suspicion. You could argue that a mouse can never get ALS because ALS is a human disease. What a mouse can do is contain some element that's a useful biological feature of the disease, and if you can identify that, you can use it as a way of discovering something important about the disease and maybe testing some therapies specifically targeted at that aspect of the disease. So I think we have to be much more constrained. So it's very unreasonable of us to expect to look down a microscope and see a mouse that has exactly the same form of pathology that we see in our human patients. This applies to Parkinson's, to Alzheimer's, and to ALS. So there's a constant problem of judging mouse models by whether they produce pathological aggregation of protein. And it's actually perhaps relatively easy to do that if you load the system enough to produce an overexpressed protein unphysiologically. But that can be misleading. It doesn't lead to translation. So it's back to my original point, which is not every aspect of the disease needs to be present or could be present in a mouse. And the only question of interest is the model useful? So here's the example of the G93A mouse, which has dominated drug discovery for the last 20 years. And there are still drugs coming into clinical trial now, which are based almost solely on SOD1 preclinical experimentation, despite the fact that there have been many, many tens, if not hundreds of examples of drugs that have failed through this route. People continue to use it. And this is a mouse in which 25 or 30 copies of the SOD1 cDNA with a G93A mutation have been inserted randomly into the mouse genome. And on the sort of appealing front, this is a mouse that gets a motor disorder, which can be measured, which has a relatively reproducible phenotype. And that is clearly very appealing and very attractive. But it gets it very early on, and it gets a very rapid and catastrophic sort of progression, which is not at the pace of change that one sees in humans, which is measured obviously over months to years. And there is a huge range of phenomena, both at the cellular and the molecular level, that can be identified associated with this mouse. And these phenomena have been used to draw conclusions about the pathways that are involved, the cell types that are involved, the degree of cell autonomy or non-autonomy in the pathological process, and ultimately to test drugs. And the problem really is that in the short life of this mouse, there's almost certainly something very interesting to be identified about why SOD1 misfolding leads to ALS. But there is a flood of severe and catastrophic phenomena which are used as readouts, which have obscured this. So one way of looking at a disease like ALS, and those of you who practice clinically in an ALS specialist clinic will always wonder whether the patients that we see, even with the mildest disability, already have a sort of end stage process, that they have gone through a period of compensation and decompensation, and that all we're seeing really is the most therapeutically challenging end stage of the process. So we assume our model is that people who get ALS have a susceptibility phase in which they are entirely normal. We don't know how long that lasts, but let's assume that it goes into adulthood. And then they have a period of dysfunction of the nervous system, which at the moment we are still trying to learn how to measure, but in which there's some compensation because there's intrinsic reserve because of the architecture, but also compensation because of functional compensation. And at that point, there is no sense in which the patient perceives themselves as having anything wrong. They don't present to the neurologist. And when they finally present to the neurologist, that period of compensation has been used up. That may just be true in a particular anatomical compartment, but it rapidly spreads to involve other regions, suggesting that each of these regions of the body is already in a state of loss of compensation. So we are studying in our human patients the phase of the disease where compensation has been used up, where the intrinsic attempts to balance the genetic mutation, if there is one, or what other processes are driving the disease, have all been used up. And so we simply then try and apply our therapeutic trials to this late phase. So we need a model that studies the quiescent preclinical phase of disease. So having a model like the SOD model, where within a few weeks of birth, there are major abnormalities going on, is intrinsically misleading and unhelpful. And so here is just one example of a recent paper from Jochen Prain's group in Dublin, where they took all the currently available models, both the SOD model and a TDP A315T model, which is also both based on overexpression, and a severe FUS model, and Riluzole, which I think we would all agree has some therapeutic benefit, even if modest in ALS, does not, in any of these mouse models, Riluzole does not have an effect on survival or anything else. And this has got to be a serious concern if we can't back translate, if you like, to say that these models have any meaning. Now, either these models don't respond to Riluzole because they are simply the wrong models. That's one possibility. But I think it's probably also the question that's being asked is not the right question. If you try and simply look at survival, for example, you will have major problems in understanding whether a drug's working, because mice die for different reasons. There are many different things that can modify the end stage of a mouse's life, and there are many drugs that do it, and these are false leads because they don't do the same thing for humans, or they're being applied far too late. TDP-43, of course, is central to the disease in the sense that it classifies our patients as having a TDP-43 proteinopathy. So any model not based on TDP-43 is unlikely to treat very many people, except in the personalized medicine context of SOD1 and FUS, where you might have antisense trials, for example. So what did we do? We created a mouse which has a human large genomic fragment with all the regulatory regions where the human promoter is driving the expression of the disease, where the disease is relatively mild and slowly progressive, and where there is a preclinical phase where the mouse appears to be essentially normal. And then we studied the cells in that mouse. You can culture primary motor neurons from the mouse, and you can find some abnormalities that point to particular pathways, very specific pathways to do with oxidative stress. And we have developed a drug discovery pipeline based on using these ES cell-derived motor neurons to try and identify whether there are particular drugs that will modify that specific pathway. So the point I'm making here, this may or may not end up being successful, of course, but the point I'm making is that the approach here is to try and create a biological system in which you can ask very targeted questions about pathways and not look for phenotypic readouts like survival, which ultimately will mislead you and lead to extremely expensive and futile human trials. So in conclusion, I would argue that the very large sums of money, and it is billions of dollars, pounds, and euros that have been devoted to studying mouse models of ALS, could have been applied in more productive ways. Maybe not initially, maybe we would have all done the same thing, but people have been locked into a way of studying these things over the last 20 years, which has hardly deviated at all, and which really fundamentally has to change, because no drug of any benefit to our patients has arisen from any of these studies. And I think people will argue that we should just keep carrying on as we are. But my argument is that we have to apply mouse models in a very different sort of a way and up to now they've been applied incorrectly and wastefully. So the specific question I was asked to address, has a study of mice been detrimental to developing therapy for ALS, I think is true, because of the resources diverted towards the wrong kind of therapeutic trials, because of the wrong kind of mouse models. And we need to do better. Thank you.